Hello, all. This is Kristen Haberther welcoming you to this Bite Size Bio webinar, which today is sponsored by GenScript. GenScript is the leading DNA synthesis partner for basic life science research, providing laboratory reagents and services to scientists from over 100 countries. GenScript offers comprehensive CRISPR services, including synthetic CRISPR sgRNA, gRNA Cas9 plasmids, single stranded DNA, purified Cas9 proteins, as well as cell line generation to help make your genome editing easy. Today's presentation is titled How to Use CRISPR to Accelerate Cancer Therapies and is being presented by Theo Roth, an MD PhD student from the University of California, San Francisco, where he is currently working with Dr. Alexander Marson. Theo studied at Stanford and did research at the National Institutes of Health before coming to UCSF to further develop gene editing tools in human T cells for cancer and autoimmune cell therapy applications. As always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll go ahead and put them to Theo at the end. The recording of the webinar is going to be available at http://bit.ly/T-cell_HDR reengineering. And I will have that link in our chat box section, which is also in your screen for GoToWebinar. So now over to you, Theo, for the presentation.
Thanks, Kristen, and thanks to the sponsors of the webinar, GenScript, for having me on today. My name's Theo Roth. Like I said, I'm an MD PhD student at UCSF, and today we talk a little bit about some of our work developing new methodologies to engineer human T cells with the end goal of developing new cancer therapeutics, but also a little bit more generally about the general types of and methodologies that are available to the broader field right now as to the ways to do targeted gene modifications, especially with a focus on therapeutic modifications of T cells, and focusing a little bit on the nature and type of DNA templates used to deliver that new genetic information into the cells. So let's just go ahead and get started. And the magic of these systems is that we can edit the genome with a power and simplicity and a speed that was never available to the pioneers of gene and cell therapies over the past 20 or 30 years. So as a quick background, a targeted nuclease system, of which CRISPR-Cas9 is the most widely used. It is made up of two components: a DNA nuclease that is targetable, wherein it can cut a target sequence of DNA, but the target sequence that it actually cuts is determined by an RNA component, which allows for rapid retargeting of that protein nuclease. I can certainly attest from running experiments in the lab that this has just changed just about everything in the nature of how we can design and test experiments around gene editing, and has really been the media hype is not overdone. This is the breakthrough that is enabling the next generation of cellular therapeutics. These two components are combined together into a functional particle that we call an RNP, or ribonucleoprotein. And this functional particle now just needs a couple things. It needs to get into the cell, and if we now are seeing evidence of gene modification, then we just need to figure out, well, what's the best way to formulate these? What are the best reagents for our specific applications? So on the first side, the delivery question of how you get these editing reagents into the cell, our lab is pretty set on using DNA electroporation, which allows for delivery of large biologic macromolecules through a fairly simple process. But there's a variety of other methodologies that may make sense for a given application, especially working on the research side in cell lines, delivering DNA components, encoding these editing reagents, and just getting the cell to express them is what's probably the most common approach. But I think especially for the research and corporate groups that work with primary cells, there's a fairly strong consensus that electroporation-based delivery of recombinantly produced protein and RNA components into the cell is the most efficient and simplest, and kind of the methodology that I think the field is coalescing around. And so, the structure of the talk today that I'm going to discuss is going to be essentially primarily is all using delivery of recombinantly produced protein and guide RNA into the cell by electroporation. So once you've settled on a delivery platform that works best for your given system, it's just a matter of choosing what's the best quality editing reagents. So that can be different types of nucleases. Again, Cas9 is the most common one, but there certainly now are a litany of other targetable nucleases that have different PAM specificities, have different fidelities now. We're going through the same process with nucleases that we did with polymerases over the past 20 years. You'll see the high fidelities come out, and then the super high fidelities, and on and on and on. The type of the guide RNA and the sequence of that is obviously quite important. But choosing if you have options of multiple guide RNAs to choosing the one that has the highest on-target specificity versus off-target effects, the stability of that RNA through various RNA modifications. And then I'm not going to talk so much about those two. They're well-worn topics in the literature. I think the area that we'll comment on a little bit today is the nature and type of a DNA template that is used during the editing process in order to insert in new pieces of genetic material. And we'll talk about a few different types and go over kind of the benefits and drawbacks of different ones. So with that, the outline of the way that we edit T cells in our lab is isolating and stimulating primary human T cells from healthy donors. The Cas9 protein and guide RNA are both recombinantly produced. We complex these together, just a brief incubation will form a functional complex. And then, by adding in electricity or putting these together in a well, we can end up with a very quick process. You press a button, it electroporates, and then you're done. You can get a heterogeneous population of cells with a variety of gene modifications. Our goal for all of this is always to get 100% knockout of our target gene. But I can say that you never get 100%, but we can certainly get, at least in our optimized primary human T cell system, well over 90, 95, 98, 99% knockouts, depending on the target site. And you can say from adapting the system into a variety of other cell types, this electroporation-based approach of RNPs can be very, very efficient across cell types and fairly quick to optimize for new cell lines or new primary cell types as well. So just to show some example data, one of our favorite target genes is CXCR4. It's a HIV co-receptor on the surface of T cells. We can, this is just some surface staining here, showing you that at least at the protein level, there's close to 99%, if not higher, knockout of this protein. Now, primary T cells might not be the only thing that you're interested in, although that certainly is the focus for cancer immunotherapies right now. But this just a brief example of how this RNP-based approach based on electroporation is widely adaptable. We've also developed a simple analogous version of that protocol for primary human B cells as well, and we can also generate high quality knockouts, as you see on the right here, knocking out an FC receptor on the surface. This is just one of many cell types that we've done these knockouts in, and you can find in the published now just about any primary cell type, and again, just about any cell line has probably there's published protocols for RNP electroporations, and certainly many of those are exceptionally high knockout percentages. Now, knockouts, though, are an exceptionally powerful and new tool on the research side, but and also the first clinical trial, at least in the U.S., focused on CRISPR-Cas9 editing, being run out of UPenn right now, is focused on knockouts of a few target genes in CD19 CAR T cells that might have therapeutic benefit. Now, but there's only so many genes that you can knock out, and two, we can only expect that knockout of so many different things in the genome is going to have therapeutic effect. Much more power comes from being able to not just delete something from the genome or remove a pathway, but actually to go in and edit individual sites and insert new DNA at specific target sites, such that now you have control over some of the programs that T cells are using, or any other cell type. And then you can design a new therapeutic function or new therapeutic program, as opposed to hoping that knockout of any given gene is going to have your desired therapeutic effect. So thankfully, inserting DNA using this electroporation and RNP, CRISPR-Cas9 RNPs, is actually quite straightforward. It just means that we have to include a DNA template in our electroporation when at the same time as we electroporate the cells in the RNP. So inclusion of a DNA template, and we'll talk about the design of those in a second, but that means that now in our bulk edited population at the end, now we have some cells that are going to be knockouts, primarily through the NHEJ or non-homologous end joining pathway. Some cells that will be knock-ins that will have the DNA template inserted at the target site, and then still unedited cells. And of course, as long as you're editing at an autosomal locus, you'll have every combination of these in cells, in individual cells, 2 alleles. But especially if the insertion that you put in is something that you can see is selectable for, sortable for, now we can actually find the cells that have a desired insertion. And so just quickly running through, I mean, a lot of the applications of these kind of approaches go without saying, but for knockouts, at least in our lab, we kind of focus on this, on new discovery approaches for fundamental biology in healthy cells, and then also in diseased patient cells, potentially modifiers of that disease or phenotype. When we can add in knock-ins, though, especially if the kind of the current most commonly applied knock-in approach using very small, short, single-stranded DNA, or also termed SSODNs for single-stranded oligonucleotide donors or donor nucleotides, now you can go in and insert small pieces of DNA, and that may be for diagnostic purposes, again, in the lab, in healthy donor cells, being able to insert a SNP or a mutation or a variant of unknown significance at a specific site and testing its functionality, or in diseased patient cells that could be going in and actually correcting an individual mutation. But from a therapeutic side, though, we'd like to be able to insert much larger pieces of DNA, and that moves our capability from just correcting mutations up to rewriting the actual genome and putting in new therapeutic functionality. And so a couple ways that have been done, the most common large knock-ins that have been published now by a variety of groups involve integrase deficient adeno-associated viruses that encode an homology-directed repair template within their genome. But I'll show data today that says that viral step is not 100% necessary, and that you can use either single-stranded or double-stranded non-viral DNA to also accomplish these similar knock-ins, potentially with a higher speed, lower cost. And then this can be used for therapeutic engineering to make a desired cell product, and whether that's for cancer immunotherapy or autoimmune disease, or for any variety of other approaches. So hopefully I've convinced you of the broad applicability and utility of being able to insert large pieces of DNA into a T cell. Now, ideally, though, when we think about, in a perfect world, what is the nature of the DNA template that we can use, again, to accelerate the development of these cellular therapies? And again, I just told you, we'd like to be able to insert very large sequences. That gives us much more control and power over the therapeutic function of those cells. Obviously, we'd like the cells to have high viability. Dosing patients with hundreds of millions, if not billions, of these modified cells. So we'd like to, have a lot of them at the start. Something that's, I think, growing in appreciation over the last year and a half or so is that we'd like these insertions to be at a targeted site. The hallmark, the foundational technology in cellular gene therapeutics, viral infection and viral integration of a DNA template, which has led development of the field for the last 30 years, is entirely based, though, on random integration of a target cassette. And I think, especially with the growing power of these targetable nucleases, and now that it's possible to integrate DNA at a specific site, then there's a growing understanding and appreciation in the field for the types of applications that are enabled by integrating at a specific locus, as opposed to integrating randomly and just overexpressing off of a viral promoter some gene. And so it is a very fun time to be thinking about these types of applications. But our techniques to insert our DNA should essentially now be able to put DNA in at a targetable site, and that's both from a quality control and QC concerns, for especially at the clinical manufacturing for cells that are going into patients, and also from the engineering side that this enables more complex and more powerful engineering approaches. But another few things that you might want to be able to target specifically with very low off-target effects, and obviously off-target effects of the guide RNA cutting are present in these systems as well, and there's a large appreciation for those, but there's been much less work done on the potential off-target effects of these newer types of targetable DNA integrations. Although from a general conceptual standpoint, these techniques are replacing random integrations of viral vectors, which of course have no specificity for a target site. So, at least from all of the patients that have been dosed with lentiviral or gamma retroviral based modifications of T cells, and of course, at least the experience of the field so far, so there's not been any reported cases of a T cell based oncogenic transformation based on those. So there's probably a little bit less concern about off-target integrations of DNA components. But just very quickly at the end, it'd be nice if this was easily multiplexable. I can tell you, one thing I didn't add on here, but it's a huge concern for doing this in the lab and then eventually also in the clinic, is how easy it is to create and generate your DNA templates. Viral templates take a lot longer than generating a non-viral template, and that just means that it's much faster to do this non-viral work. And that's what the kind of the point is at the end, that this should be a rapid process, so that we're not going to find the best way to modify T cells by all sitting around and coming up with one construct that everybody's going to test. These are a process of trial and error, and we're going to have to it's going to be a matter of testing hundreds, if not thousands, of different constructs and finding the ones that have the therapeutic properties that are most applicable to a given therapeutic or research setting. And the faster that we can insert and test constructs, the faster we're going to work through all of those ideas and find the ones that are best suited. So so these are all things to keep in mind when thinking about, and this isn't to say that there's any one the best way to do these, but individual applications will always have different considerations. So when we a few of the types of DNA templates that are I think currently available, or current options. One that we had previously published on in our lab a couple years ago is using a short single-stranded piece of DNA. This is can be chemically synthesized, just like a DNA primer is synthesized, and it can be shipped to you in a week. Now, the issue is that because we have to include homology arms to target the integration of whatever new piece of DNA that you want, the limitations of DNA synthesis end up being about 200 base pairs. And so that means that you essentially, if you're including homology arms that are about 80 base pairs long, there's only a very small amount of DNA that you can insert. This is certainly more than enough for mutation correction applications. But for larger scale engineering, we'd really like to be able to insert much larger pieces of DNA. And the three ways that I think there are to do that include a much longer piece of single-stranded DNA, using a viral genome. Again, integrase deficient adeno-associated viruses are the most commonly used one, but integrase deficient lentiviruses or adenoviruses have also been used in published studies. Or finally, a piece of just a piece of non-viral double-stranded DNA can also serve as an end plate. And these three all have the capacity to insert much larger pieces of DNA. And now, when you can insert a much larger piece of data, that actually makes the experiments a little bit easier because it makes the readout of whether a cell got a targeted insertion a little bit simpler. So here's the design of just kind of a standard homology-directed repair template. They include a gene payload, whatever the new piece of DNA that you want to put in. In this case, it's just a GFP fusion tag. And then on either side, a homology arm, perfectly homologous to the DNA sequences around a given cut site for a guide RNA. So in this case, we have a guide RNA cutting at the RAB11A locus. It's just a highly expressed housekeeping gene. And the guide RNA comes in, it cuts right there. The cell notices that there's DNA damage at that site. It can repair that damage via one of two pathways, either non-homologous end joining, just slapping those two exposed ends back together and religating it, which is sometimes an error-prone process, which leads to the frame shift mutations and subsequently protein knockouts that so many of us now use as a mainstay manipulation for research purposes. But there is a second repair pathway known as homology-directed repair, which is when a cell, as long as the cell is going through cell cycle, the cell has a chance to use the other chromosome, especially when the chromosomes are lined up, again, as the cell progresses through cell cycle. It can use the other chromosome, which is homologous in the regions around where that double-stranded break is, to do a much less lossy DNA repair process, whereby the other chromosome is templated off of to repair that strand, and then through a lot of complex crossing over, then you end up with a perfectly repaired strand. Now, if you confuse the cell and you have an HDR template that has these long homology arms, the cell thinks that it's crossing over onto its other chromosome, but it's actually onto your template. And you can insert, and if there's any new DNA in the middle, the cell's none the wiser, and it just integrates that DNA at your targeted site. The big, huge advantage of this is that it gives you total sequence control over what the final genomic sequence is, which again is a lot of fun both in the lab and for imagining therapeutic processes, because you have so much power over what the cell's genome now is, and then you can use that to re-engineer the functionality of those cells. So we went through a fairly extensive process to optimize these using a non-viral DNA template and electroporation in primary human T cells. I can tell you it was a lot of 96-well plates, testing out conditions, and some things that these four primary areas of optimization. And we just look to see, are cells getting a GFP integrated, and then how viable are the cells? Because it doesn't matter if you have perfect efficiency, 100% of one cell is not a viable therapeutic strategy. But of course, if you have perfect viability but have very low or no editing of those cells, that's also not a useful therapeutic strategy. So there's a balance between these two. So looking at this control locus here, in one of the nicer donors that we've looked at, that this site integration of GFP went well over 50% in this donor. I can say that our average at some of these housekeeping genes where we just knock in GFP, usually across donors ranges from 30 to 40%, at least with our current iteration of the protocol. So if we can target this at one site, I told you that non-viral templates, one of the real advantages is that it's very easy to make new templates and target them across the genome. So looking here at 6 different housekeeping genes or T-cell specific or highly expressed genes, we chose these 6 RAV11A, but some other ones that you might recognize: the CD4 co-receptor, clathrin, beta-actin, FBL, which is a nucleolar marker, or beta-tubulin. We chose these because they have hallmark cellular localizations, and so as a control to make sure that these GFP positive cells that we're seeing and that we're robustly generating actually have the integration at the targeted locus, we designed GFP fusion templates for each of these genes, and then by just doing some simple confocal microscopy to look to see where does the GFP go in these cells, all of these 6 images are taken from live primary human T cells that now have a GFP conjugated to their to these individual genes that have different hallmark cellular localizations. So you can see here we're labeling clathrin microvesicles, actin microfilaments, especially all the microvilli that are highly active and present on the surface of T cells, the nucleolus, the microtubule organizing center on the far right. This gave us a high confidence that these integrations are going at the desired target site, because there's no way that a GFP that's randomly expressing is going to specifically enrich at each of these different loci. So that gives a pretty high confidence, and again, going across these different targets, that both we are integrating at our desired loci at very robust efficiencies, and that these knock-ins are specific. So exploring this, the kind of the power of these knock-ins just a little bit, by knocking in at these individual sites, and maybe you noticed from the flow plots here, these different genes have different levels of expression of GFP. And the easiest one to look at is that beta-tubulin, which is in the top 10 most expressed genes in T cells and also in most cells. We can see that this GFP expression level is quite a bit higher than some of these other genes. So if we go in and look at this across donors, we can see that in regulatory control and regulatory circuits for these endogenous loci are maintained and can be taken advantage of when you knock in a specific target gene. And just a quick demonstration of that is that across 6 donors, the beta-tubulin is always by far the most highly expressed, and then actually all of these different genes end up across these donors being expressed at just about the same amount in each donor. And that across these different template sites, and so that gives again a lot of confidence that we're both integrating at the target site, and also that targeting integration to a specific site enables a lot of exciting thoughts about how to take advantage of endogenous regulatory control for given applications. So if we can knock one thing in, obviously a quick question is, can you go in and knock more than one thing in at a time? And again, one of the advantages of non-viral DNA templates is that we can make up these new different templates very quickly. So on the left, we have an example of multiplex knock-in, putting a GFP at the clathrin locus and putting an mCherry in at the RAB11A locus, and what we can see is a robust dual positive population that's actually enriched over what you would expect by random chance if these 2 processes were independent. And we have a lot more extensions of this stuff in our recent publications, so I definitely recommend you check out the supplementary figures there, because I'd love to know that somebody looked at them, and we spent a lot of time making them. So but anyway, beyond multiplexing at multiple allelic sites, we also can show that you can by putting in 2 different detectable DNA sequences at a single genomic site, then you can detect biallelic modifications as well. So now putting a GFP and an mCherry into the RAB11A locus, then we can sort out a very robust dual positive population that now you could potentially, if for research or therapeutic purposes needed to have biallelic modifications, now you can actually sort out for those cells. The percentage of biallelic modification is going to be higher. It's going to be roughly double that, because you'll also have cells that have 2 GFPs integrated and 2 mCherries integrated, but that's not something that you can detect or sort on. Um, so kind of summarizing the discussions of the technical side of our work, I think this is a pipeline that we're calling non-viral gene targeting that is rapid, efficient, and is very adaptable. It's very easy to move from one target site to another. The RNP formation is just a quick incubation. Getting the Cas9 and the guide RNA, those can each be easily purchased commercially, shipped to you in a week. On the human T cell side, isolating cells from a healthy donor or soon from patients, they just have to be isolated and stimulated using very similar stimulation and culture protocols as viral modification, as current lentiviral and retroviral T cell modification protocols. So that's a fairly well known technical side. And then finally, the DNA template. At least all of the stuff that I've shown you so far has been with a linear double-stranded DNA template. And so these are usually sequences that we have to clone together. I mean, we need to add on the homology arms and whatever we want that functional insert to be. But that cloning process is very straightforward and simple, and you can buy large pieces of double-stranded DNA for cloning applications pretty cheaply now. And so you make that, making the HTR template can be as simple as doing some large PCRs and purifying it, that DNA down. And then the electroporation is an exceptionally simple DNA delivery technique. You just mix all 3 of these components together, press a button, it shoots, and then now you have T cells that you can expand and phenotype. So, this is kind of the technical pipeline that we've developed. Now, I'll tell you 2 quick stories about how we're applying this currently, and then move into a more general discussion about different DNA template types and what the advantages and drawbacks of some of them might be. So one application that we've used this for is actually, I said that kind of using these short single-stranded DNA donors or SSODNs allows you to correct mutations. So, and that's not great if what you want to do is insert a CAR into a T cell because you need 1.7, 1.8 kb of DNA inserted. But if your application is correcting a single base pair at a targeted site in the genome, then an SSODN is actually quite straightforward and easy to use. So this is not to get too much into the biology of this, but there's a family where the 3 of the children in the family have 2 different mutations or compound heterozygotes for loss of function mutations in the IL-2 receptor alpha, or CD25, the high affinity IL-2 receptor. IL-2 is critically important for regulatory T cell function. These patients have regulatory T cells, but they are largely non-functional. And so, and if we look on the surface of their bulk T cell population, we see that there's little to no CD25 expression. We hypothesized that correcting one of these causative mutations in the patient's regulatory T cells may be a targeted, personalized cellular therapy for these patients' pleiotropic autoimmune diseases. Designing these is straightforward. The patients have a mutation at G2A. We come in with a HDR template. This can either be a large one with long homology arms or a short SSODN with much shorter homology arms. The HDR template contains the correct DNA sequence, and then it has another silent mutation to prevent rebinding and recutting by the guide RNA by removing the PAM specificity. And then, if HDR occurs, we have integration of the new targeted, the correct sequence that then corrects the function. And you notice there's a TAG created here. So there's an early stop codon for these patients. And then we correct that back and reestablish the normal reading frame. So when we actually go in and look this and use this protocol, we can see that in these patients' T cells and also in their regulatory T cells, if you dig through the paper, we can actually correct expression of CD25 on these patients' cells at a fairly high degree. And so now we're pursuing this as a personalized cell therapy using a non-viral template in these patients' regulatory T cells. Something that would requires much larger modification, though, is going in and actually modifying a much larger piece of inserting a much larger piece of DNA to do protein scale engineering. The first protein that comes to mind when you think about T cells is the T cell receptor, the receptor that gives it its specificity for a variety of pathogens or other antigens. What we'd like to do is, as opposed to current techniques to insert in a new either chimeric antigen receptor or synthetic TCR randomly in the genome, we'd like to be able to go in and actually replace just the endogenous receptor. And actually, in this case, we don't even need to replace the entire endogenous receptor. We just need to replace the extracellular portion, which confers specificity. This both allows you to use smaller construct sizes, but it also takes advantage of the endogenous regulatory control of the T cell receptor. And both in our recent publication showing non-viral T cell receptor replacement, and also a publication from Equim et al. From Michelle Sadelin's group at Memorial Sloan Kettering in 2017 in Nature, using a chimeric antigen receptor targeted to the endogenous T cell receptor locus, we've both found that there are potential advantages to targeting at the endogenous site over viral overexpression. And so that's worthy of a whole another webinar. But we'll talk about, I'll show you kind of the construct design and the process for putting in a new specificity at a synthetic receptor. So to do this, though, T cell receptor is a multi-component complex, but the two that determine specificities, the TCR beta and alpha loci. We target the first exon of the constant region of the TCR alpha locus, and I'm sorry that this genomic stuff gets a little bit complex, but this is how the T cells evolve, or how our the adaptive immune system evolved in order to have all these different specificities. So it's a wonderfully complex locus, but also means that it's challenging from an engineering perspective. But we've worked out a strategy that works quite well by inserting at this first exon of the constant locus, which allows you to target every single T cell in a polyclonal population. We insert a construct that has a multi-excision peptide followed by a full-length new TCR beta chain, a second multi-excision peptide, and then just the variable regions of the TCR alpha chain. So that now, when this goes in, when HDR actually occurs, now we see an expanded first exon that has both the entire beta chain as well as the new specificity of an alpha chain with these multi-excision peptides, which become important when we think about what happens with transcription and translation to form the final functional proteins. Upon transcription, the alpha exons splice in with their endogenous additional exons to form the entire endogenous TCR alpha locus. And then upon translation, these multi-excision peptides pop these different components apart. And so the endogenous VJ, which again, you don't know what an individual cell's VJ region that it's going to be expressing is, but that gets separated from the other transcripts by this multi-excision peptide. And now this does not include a transmembrane domain. This is now non-functional and most likely is going to be degraded. And now you have two full functional translations, a new beta chain and a new alpha chain. And so now, what the actual specificity that you knock in here is fairly arbitrary, because the efficiency of the knock-in depends on the guide RNA and the homology arms, and you can just swap in whatever that specificity that you want to be is. So just showing you that we can do this here, standing on the x-axis for TCR expression, and on the y-axis for tetramer staining for the specific TCR that we just knocked in. We can see that when we put the DNA template in, but we include an off-target guide RNA, we don't see any TCR knockout, and we don't see any knock-in of our desired new TCR. But when we include the non-target DNA or guide RNA, which cuts at the TCR locus, TCR alpha locus, we see that the majority of the cells are a knockout, and that's this large population in the lower left. But we also see a fairly robust population of knock-in cells with this new TCR. And then, this population is easily sortable or selectable. And now can be used in a variety of downstream functional applications. And these applications aren't necessarily the point. Again, you go into much greater detail about the functionality of these cells in our recent publication. But just to show you that these cells kill with essentially the same kinetics as virally delivered, the same TCR virally delivered. So this is just a simple killing assay with a target cell line that expresses both the right MHC allele as well as the right target antigen. In this case, the melanoma cancer antigen NY-ESO1. So, I told you these two vignettes about how some of the first ways that we're going in and applying these the ability to knock DNA in at a specific target site, but looping back again around at the end to what is the nature of the DNA template, and how might different types of templates be right for different applications. Again, short single-stranded DNA or SSODNs is the simplest. Because you can just plug in a sequence to any of a variety of DNA oligo providers, and they'll ship that to you next week. The limitation is that the size of the knock-in is much lower. So these other types of templates, either long single-stranded DNA, double-stranded DNA, or a viral genome, again primarily being adeno-associated viruses, these all allow you to integrate much larger DNA. But there's a variety of other considerations as well that you can think about for choosing a DNA template type. So one of these is off-target integrations. And this is something that has not been necessarily looked at that much. But if you look in both viral, so some of the papers have been published with a viral delivery or targeted viral delivery, even though you're using an integrase deficient virus, if you can study the flow plots, there is definitely off-target integration of these DNA templates. And the question is, well, is that off-target integration of DNA templates both interfering with your conclusions from the template, and also is it potentially a therapeutic concern? And I think that considering the fact that now thousands of patients have been dosed with lentiviral or retroviral delivered DNA templates in therapeutic trials, both for CAR T cells and a variety of others, without any reported cases of oncogenic transformations, we have to compare it against that with these targeted integrations, or the potential off targets when using these targeted DNA templates, with the fact that every clinical application so far doesn't even have the concept of a targeted or of an on-target versus off-target integration, because they all integrated off-target sites. They're random integrations. So that says that it's probably not a big concern. But that's something that definitely needs a lot more work to be conclusively determined before these new targeted integration methodologies are used in the clinic. But here, looking at this, is using a double-stranded DNA template, a matrix of 6 different templates, and then plugging in those against 6 different guides, and then one control guide and then no guide. We see that essentially by far the highest GFP expression comes when using the correct guide. But if you have a good discernment for shades of blue, you can see that there is a little bit of off-target integration for some of these combinations of an off-target guide and a DNA template that is not targeted to its specific locus. Looking at this in just one case, again, we only see efficient integration of GFP when we use a DNA template with the on-target guide RNA. But when we electroporate either the DNA template by itself with a scrambled RNA that doesn't guide RNA that doesn't cut anywhere, or especially with an off-target guide RNA that cuts somewhere else in the genome, you can start to see a population, an observable population of GFP positive cells. And this is by no means every single cell that might have gotten a DNA integration, but the fact that there is a population that is observable means that we can now do experiments around that to try to minimize the off-target events and see if any of these different DNA template types are better or worse in that regard. And so just to show a little bit of data, again, this is with double-stranded DNA template. We can see a small amount of off-target integrations when we use a guide RNA that efficiently cuts somewhere else in the genome. Again, the DNA template by itself has much lower integration, but comparing this with a long single-stranded DNA template, we actually see a quite noticeable, reproducible, and fairly large reduction in this observable off-target DNA integrations using a single-stranded DNA template. Again, summarizing data across quite a few donors, we see that on-target insertion efficiencies, whether using a double-stranded or single-stranded DNA template, are quite comparable, but that when doing a variety of off-target assays using an off-target guide RNA or just delivering the DNA template by itself on the left, we can see that the long single-stranded DNA templates can actually reduce the observed off-target integrations. Looking at these off-target integration profiles, these long single-stranded DNA actually is potentially ideal in that regard. But again, there's still a lot of testing to be done to see whether that's actually a concern or not, both on the research side and therapeutically. But both viral delivery and double-stranded DNA have the issue of off-target integration, have observable off-target integrations. And again, compared to lentiviruses where every integration is an off-target integration, then these are certainly much better. But again, we're trying to differentiate between the different types of DNA templates. So there's the issue with long single-stranded DNA is essentially entirely in the production of it. It's quite difficult to make large amounts of long single-stranded DNA, especially at the very high concentrations necessary for these electroporation approaches. We've now published 2 different approaches to making this single-stranded DNA. On the left, there's an IVT or in vitro transcription followed by reverse transcription based approach developed by Manu Leonetti, a fellow at the Chan Zuckerberg Biohub here in San Francisco, which uses first an in vitro transcription to make a single-stranded RNA template off of a desired DNA HTR template, followed by reverse transcription to make a single-stranded DNA, single-stranded RNA hybrid, which can then the RNA can be selectively degraded, and now you have single-stranded DNA. The other approach is to use selective end modifications on a double-stranded DNA template. In this case, this is using an exonuclease that is specific for a 5' phosphate to then degrade selectively degrade one strand. I can say both of these can be done, hands-on yourself in the lab, but as someone who's done this a lot, they are fairly time-consuming and a little bit laborious, and the enzymes can be a little expensive. So the big issue with these single-stranded DNA is in the ease of production. Fortunately, though, there's now a couple different companies have recognized that this is a bottleneck in some of these these cell therapy and engineering applications, and now there are commercially available long single-stranded DNA constructs or products available at the very high concentrations necessary to insert them. And I can say that we've used one. The sponsor of this webinar, GenScript, has a new product. They're not the only company that does this. I know IDT and at least 1 or 2 other companies also make long single-stranded DNA templates. But we've specifically used the GenScript product before, and it worked quite well in our hands. So very essentially comparable to the single-stranded DNA that we were formerly making in-house. So it is now possible to purchase this type of template as well. But viral genomes, they're a little bit harder to produce because you have to make a virus. The easiest things are either the short single-stranded DNA, which you can current just buy today, or double-stranded DNA, which is easy to make by PCR. A variety of other issues with toxicity. We've noticed that single-stranded DNA has lower toxicity issues than either the viruses or double-stranded DNA. And then also a potential therapeutic advantage is types of templates that do not have to go through a viral vector. That has the potential advantages at a number of steps, but especially both in the simplicity and ease of production. It's producing viral vectors for therapeutic at GMP quality and for therapeutic use is such a huge issue that there's been New York Times articles about this. So these arcane production issues are such a bottleneck for the field that it enters the popular press, and so that's certainly a good argument for why alternative, easier types of templates are needed to move these cell therapies forward. And then it also gets to the final question of if your application is the actual end GMP production of cells, then the availability of these reagents at GMP quality is a crucial concern. So I'll leave you with this non-viral gene targeting is a rapid, highly adaptable, and simple protocol for integrating large pieces of DNA at specific target sites in the genome of primary human T cells. And this is a technique that we think is going to be central to accelerating the design, testing, and construction, both on the research and side and therapeutic side of gene-modified T cells for cancer immunotherapies and hopefully a variety of other applications as well. So I will quickly acknowledge some of the people that helped us out. We're working with a large number of groups, but especially everybody in the Marson Lab and a few key collaborators, Manu Leonetti at the Chan Zuckerberg Biohub, Jennifer Doudna at the Innovative Genomic Institutes and UC Berkeley, a variety of clinical collaborators on the CD25 story, and especially Tony Ribas and Christina Saus-Puig at UCLA, who've worked with us on the TCR replacement application. And also, of course, the sponsors of this webinar, GenScript and Bitesize Bio. So with that, that's the end of my slide. I think GenScript has a few slides that they'll present as well. But if you have questions on gene editing or any of the techniques or methodologies that I discussed, please feel free to reach out. My email's at the bottom. You can there's a lot more detail in our recent publication, and happy to field questions from you guys now or by email in the future.
Great, Theo. Thanks so much for that presentation. That was really, really interesting, and I'm super excited to get into these questions. But like you said, before we do get audience questions, which, as I had mentioned at the beginning, if anybody does have any questions, you can go ahead and write them in the questions box to the right of your screen, and we will get to them in just a second. So we do have a few messages from our sponsor. If you could advance, Theo. GenScript aims to make genome editing easy for you. GenScript provides basic CRISPR reagents such as gRNA Cas9 plasmids and the RNP system. Our gRNA Cas9 plasmids are pre-validated by the Broad Institute of MIT and Harvard, and we provide both the dual vector and all-in-one vector system for sgRNA expression. In addition, we also have SAM constructs for transcription activation. For the RNP system, we provide fully customizable synthetic sgRNAs, unmodified or modified, as well as synthetic custom cRNAs and tracer RNAs. We also provide SP-Cas9 nuclease with 2 NLS for optimized nuclear compartmentalization and Cas9 protein with 1 NLS and an EGFP marker allowing for fluorescence activation cell sorting. Many studies, including the studies that Theo just presented here, demonstrate that single-stranded DNA are the best form of HDR DNA donor templates for CRISPR-based gene insertion. GenScript is proud to announce that we are also offering single-stranded DNAs that are up to 8,000 nucleotides long in length to serve as HDR DNA donor templates. By using our proprietary harsh chemical-free enzymatic approach for producing single-stranded DNAs, we are able to offer high-quality, sequence-verified, single-stranded DNAs that are up to 8,000 bases long in length. We also offer single-stranded DNAs with up to 40 micrograms delivery quantity, allowing for flexible study design. For more information, please visit our website listed here. Slide. We also provide various technical resources that can help make your research easy. For example, you can check out our new CRISPR RNP user manual or our comprehensive CRISPR handbook for free. Slide. Finally, we have a great 2018 fall webinar series lined up with 7 fantastic topics. The first webinar shown the first 4 webinars shown here, which include the one you just attended, can be found on our website for on-demand viewing. Registration is still open for the last 3, which can be found at www.genscript.com/webinars. We hope you find these resources helpful and have great success with your research. So we do actually have a few questions from the audience. I'm going to go ahead and pose these to you, Theo. And again, if anybody continues to have questions as we're speaking, please feel free to put them into the questions box to the right of your screen. So first one is, in your paper, did you use single-stranded DNA or double-stranded DNA for integration into TRAC?
Yeah. So in our published paper, you can dig through the legends for each individual experiment. But the TCR integrations that we did were using double-stranded DNA.
Okay.
Double-stranded DNA has the advantage that it's very rapid to produce and easy. We have done the PCR integrations with using single-stranded DNA, actually using the GenScript's single-stranded DNA product, and we see very comparable integration efficiencies.
All right, great. That was a question from Justin, and they also say awesome paper, by the way.
Right.
So I actually have a question for you. In your clinical study where you were looking at the mutation for IL-2 receptor alpha chain, do you know, or are you guys planning on looking at how long that mutation correction actually lasts, like doing longitudinal
Yeah.
studies?
Yeah, well, that's certainly going to be a focus of the clinical trial that we're currently putting together for those patients. Anytime when you're editing in a non-permanent cell type, of which T cells can be
Mm-hmm.
very, very long-lived, but they're not the equivalent of hematopoietic stem cells, which
Right.
you would expect to be a lifelong cellular therapy. We have the question of how long is our therapeutic product going to be present in the patient.
Mm-hmm.
I can say from regulatory T cell based clinical trials that have been pioneered here at UCSF through Jeff Bluestone's group in the past 5 or 6 years for type 1 diabetes. They found that adoptive transfer of regulatory T cells, adoptively transferred regulatory T cells can persist in a patient for well over a year.
Mm-hmm.
And actually, essentially, it's been as long as they've looked. So we're cautiously optimistic that that some of the corrected T cells will be long-lived, and also that the ones that are long-lived will be the ones that are binding or that are recognizing autoantigens, and may very well be the most important ones to have around for a long period of time.
Yeah.
But of course, it's something that it will be a key part of the clinical study.
Absolutely. Yeah. It's can get a little touchy with putting all those cells back in the patient for sure.
Absolutely.
Um, One question we also have is, um, this is from Takuya. What is the maximum length you have succeeded in insertion? What's the maximum insertion length you've gotten?
So we've tried larger and larger templates. I can say that without going too much into the details, but we've now inserted things that are a good degree longer, many kb. What we find is that as you increase the size of the DNA template, that the efficiency does start to go down a little bit. And this might be something that's a little bit different with viral DNA templates, because larger integrations using integrase efficient viral templates appear to be of similar efficiency as smaller ones. We think that this is a mass versus versus molarity issue. As you increase the amount of DNA that you can deliver into a cells
Mm-hmm.
is limited by DNA mass that we find. The efficiency of integration is dependent on the molarity of DNA. More
Sure.
copies
Mm-hmm.
of DNA, the higher efficiency is.
Right.
So as you get to larger and larger templates, for the same amount of mass, you have fewer moles, and so we find that the efficiency starts to decrease a little bit.
Yeah, no, that makes sense. All right. Um, One more question is, when you had your insertions, when you were when you were looking at making um, your TCR, you had
Mhm.
included both P2A and T2A. What was the difference?
Yes. So yeah, the
Why did you put
two-way
them where you
sequence.
put them, I suppose?
Yeah, yeah, okay. So I hope I understand the question correctly, and please,
Yeah.
just ask again if I am not getting at the specific point.
Hmm.
But these two-way sequences are one of many little tricks that viruses have, wonderful little tricks that viruses have evolved in order to have very, very compact genomes.
Hmm.
We take advantage of these because we have essentially the same problem. We have a very small we have limited real estate in the DNA sequence that we can integrate. So we want to pack as much stuff as we can to get the desired functionality in as little DNA sequence as possible. Two-way sites or also like multi-cistronic sites
Hmm.
or self-excision peptides are a short sequence about 20 amino acids long that when the ribosome runs over that, when the ribosome translates those 20 amino acids, it actually causes a break in the polypeptide chain, which then allows you to have multiple separate cistrons or protein products off of a single promoter.
Hmm.
Um, using different if you have, so you have one two-way site can give you two different polypeptides or proteins.
Hmm.
If you have multiple two-way sites, now you can get up to any arbitrary number. Um, the there's a variety of different versions of this sequence isolated from different viruses. P2A, T2A, F2A, E2A. There's a whole bunch of other ones as well. Um, Using the different sequences, though, there's no necessity to
Hmm.
have different sequences. But when you're cloning a DNA construct, it's not desirable to have highly homologous sequences at multiple points
Sure.
in the sequence of DNA. So you can use multiple different sequences to make your cloning easier, and they all function about the same.
Gotcha. All right, good to know. And then as of right now, so the last question is, what is the cost of double-stranded DNA versus single-stranded?
Yeah, so that's one of the advantages of using double-stranded DNA is that it's faster to make and cheaper to produce than the single-stranded DNA is, and a little bit easier to produce at very high quantities, at least right now.
Yeah.
Making double-stranded DNA is as simple as doing a lot of PCRs. So I mean, you got to pay for a high fidelity PCR enzyme,
Sure.
and you have to pay for whatever the DNA purification methodology that you're going to use is. We're a big fan of magnetic bead based DNA purifications or
Oh yeah.
spry purifications, um, but those two together actually make double-stranded DNA quite inexpensive.
Yeah.
Um, Single-stranded DNA, because it requires enzymes that are not produced at such high volumes, is currently more expensive. But again, as we discussed, there are unique advantages to it as well, though.
Perfect. Yeah. Well, thank you again for giving this really, really interesting webinar. I'm really excited to see how things progress. It seems like it's going to be a very useful tool for all sorts of diseases, cancer, viral based, HIV, all of it. So
Well, we certainly hope so.
oh, wait, there's a couple more questions that just came in. Sorry.
Great.
Okay,
Happy to.
how much troubleshooting, Darren wants to know how much troubleshooting went into choosing 300 base pair HAs?
Yeah, we did a little bit of optimization around that. We found that 300 base pairs was a great compromise between efficiency, again, that mass versus molarity effect.
Hmm.
As the homology arms get longer, you for the same amount of mass, you get fewer and fewer copies of the DNA template.
Right.
And so you know, don't necessarily have conclusive data on some of these things, but the trade-off that we ended up with settling on about 300 base pairs was that kept the size of the constructs down a little bit more, as opposed to using 1 kb or 2 kb homology arms, which are common when using like plasmid donors for like mouse zygote applications.
Hmm.
Um, um, but also, those longer homology arms were much more efficient than using the shorter, like 60 or 80 base pair homology arms, which is the max that you can get away with for the SSODNs.
Gotcha. Okay. Um, and how much RNP slash template DNA were used to perform electroporations of 100 million T cells?
Yeah. So, so the numbers that I know off the top of my head are based for our standard electroporation, which is just 1 million T cells. So we've got all this stuff in the method in our methods section
Right, on your
of
paper.
our
Hmm.
paper, so you can dig in if you want to get a real nitty gritty. Um, but for a standard 1 million T cell electroporation, which we use the Lonza 4D system,
Okay.
which has a 20 microliter electroporation volume. So we have 1 million T cells in that, and then we usually electroporate about 50
Okay.
picomoles of RNP
Hmm.
in that. That's usually concentrated such that that takes up about 2.5 microliters of volume. And then into that, we electroporate about 1 microliter of a DNA template, which is, depending on the template, usually between 1 to 3 or 4 micrograms per microliter. And that's definitely something that you can optimize for a given template, is the total amount of DNA to electroporate. So it's about it's on the microgram scale for
Hmm.
DNA templates, and then about 50 picomoles of RNP on the RNP side.
All right, cool. Yeah, that makes sense. Justin asks, so it's not a two-part, but it's a question and then an explanation. So they ask, did you sequence your T cell TRAC locus before designing your HAs? I ask because the reference sequence that I find differs from your published HAs.
Yeah, so we pulled the sequences from so there's two things. We pulled the sequence from the published human genome, um, like HG38 build. Um, the we There are one or two silent mutations integrated in there to abrogate guide RNA binding sites.
Hmm.
So that will cause slight differences, but we've certainly found, and others have found, that a small number of mutations within a homology arm does not affect the integration efficiency.
Hmm.
Um, so beyond that, you know if during all the cloning steps and stuff, we validate the sequences and things to make sure that it was as we originally designed, but there were some small differences um, that were inserted inserted at the design stage.
Sure.
And there is a more subtle discussion that I didn't talk about today, but we've been able to insert new DNA at both the TCR alpha locus, as well as do an analogous construct at the TCR beta locus as well,
Oh.
or even to multiplex this, where you replace just the alpha locus at TCR alpha and just the beta locus at TCR beta.
Uh-huh.
TCR beta gets a little bit more complex because there's two copies of the constant domain,
Right.
and so essentially we had to choose one of those two copies to serve as our homology arms. And
Gotcha.
so there is a for those TCR beta constructs, they are specific. The DNA sequence matches I forget which one off the top of my head, but TRBC1 or 2. But we actually found that that sequence works for both of them. So.
Oh, wow. That's really interesting. Very cool. Justin says thank you. He understands. Or they understand.
Great.
Um, great. Well, thanks again, Lee. Thanks again, Theo, for answering everybody's questions and giving such an illuminating presentation. We really appreciate it. And thanks also to our sponsor, GenScript. Finally, thanks to everybody who attended and took the time to ask questions and listen in. If you've enjoyed this seminar and would like to view some other video recordings of the session, you can visit, or if you would like to look at this again, my apologies, you can go ahead and visit our webinars page at bitesizebio.com. It should be up in the next 24 hours. And you can also view some other webinars that we have lined up for you at Bitesize Bio, at least for the rest of the year. So until next time, good luck with all of your research, and goodbye to all of us at GenScript and Bitesize Bio.